All right, this next series of slides has us looking more deeply into sex-linked traits. And so here we're looking at the X and the Y chromosome side by side, and you can see there's a huge size difference between them. And they are not really homologous chromosomes. There are some homologous genes on them, but very few. So of the 1,098 genes that are on the X chromosome, there are only about 30 on the Y chromosome that may cross over with the X chromosome during meiosis. But the X and Y are the sex chromosomes, as opposed to the autosomes. All the other chromosomes are referred to as autosomes, if you'll recall. So they determine biological gender. The X and Y chromosome determine biological gender. And I'm specifying biological gender here because, as I'm sure you're aware, it can be a little bit more complicated than that in humans and even other species too. So a sex-linked gene, as you should remember, is any gene on the X chromosome with no homologous locus on the Y chromosome. Remember, locus just is short for location. So we use locus to refer to the location of a gene on a chromosome. For example, Duchenne muscular dystrophy, the gene that mutates and causes Duchenne muscular dystrophy, is right here on the X chromosome. That's its gene locus. So there's a gene locus on the X chromosome, but there's not one on the Y chromosome. And again, that's most of the genes on the X chromosome. About 168 sex-linked disorders have been identified, and that involves about 113 genes. So there are 113 genes that can mutate in different ways to cause more disorders than there are genes, if you're wondering how that can happen. The Y chromosome is much smaller and contains only a few genes. The most important is the testes determining factor, also known as TDF. So that's the only one that's shaded here on the Y chromosome. It's the most important. Of the 75 protein coding genes that are on the Y chromosome, the most interesting or significant one is the testes determining factor. It's also known as the sex determining region on the Y chromosome. So sex determining region on the Y, so it goes by SRY also. So TDF for testes determining factor, or SRY for sex determining region on the Y chromosome. So as I already mentioned, they're not really a homologous pair, not a true homologous pair, and only about 30 genes will cross over between homologous regions at their tips. So at the tips of any two chromosomes, homologous pairs of chromosomes, the very tips are the most likely to cross over. And when it comes to the X and the Y chromosome, that only happens at the very tips. And those are known as pseudo-autosomal regions because they behave like autosomal chromosomes. In other words, the genes will independently assort. Those genes will independently assort just as if they are on autosomes instead of sex chromosomes. And they go by PAR1 and PAR2 for pseudo-autosomal regions. So there's a pseudo-autosomal, or PAR1, pseudo-autosomal region 1 is at the top of the chromosomes and includes about 25 genes, whereas pseudo-autosomal region 2, PAR2, is at the bottom of the chromosome and involves fewer genes, something around 5. So that's how we get a total of approximately 30 genes. So not too much crossing over going on between the X and the Y chromosome. Not a whole lot of recombination, because there's not a lot of homology. But just like the autosomes, the crossing over that does occur, the minimal crossing over that does occur between the X and Y, happens during prophase 1 of meiosis. If you'll recall, that's when it happens. And then the last thing to point out before we move on is that we're going to be taking a closer look at, these are some of the X-linked disorders of the 168. We're only looking at a few here, and these are some of the most common ones. So we're going to be taking a closer look at Duchenne muscular dystrophy, color blindness, and hemophilia as examples of sex-linked traits. And we're going to start with color blindness. And so there are 3 genes associated with color vision on the X chromosome, and a mutation of any one of these will cause the different forms of color blindness that we see in the human population. Males are more likely to express sex-linked traits because they are hemizygous, if you'll recall. That means they only have one allele. Hemi means half. So they have half the normal number of alleles. Usually you have 2 alleles for every gene, one from your mother, one from your father. But in the case of sex-linked traits, males only have 1 gene for those genes that are only on the X chromosome. So that's half the normal number, and then we call it hemizygous instead of homozygous or heterozygous. Hemizygous, half. They cannot be heterozygous. And that's what causes them to always show the trait that is coded for by the one allele that they do have. So they always express the one allele they receive from their mother. So it's all because of their mother. So in this Punnett square, we have an example where we're crossing a father with normal vision because he has the dominant allele, with a mother that is a carrier for color blindness. She has 1 recessive allele, but she's heterozygous. Father's hemizygous dominant, but the mother is heterozygous. And we see the same 3-to-1 phenotypic ratio that we normally see. However, the only ones that have the recessive disorder, color blindness in this case, is the 1/4 that shows it are only males. So again, that's our clue that we're dealing with a sex-linked trait. Females are less likely to show sex-linked traits because they can be heterozygous and therefore carry the dominant allele that will mask or cover up the recessive allele, the effect of the recessive allele. So only females can be carriers. Men can't be carriers, or males can't be carriers of sex-linked recessive traits. But females can because they can be heterozygous, and therefore they don't exhibit like the daughters here that receive the recessive allele. They don't show color blindness. They don't have color blindness, but they carry the allele for color blindness because they're heterozygous. So that recessive allele is kind of hiding out in them, and you can't see it in their phenotype. They're carrying it, and without a genetic test or knowing the parents and then suspecting that they might be a carrier, they have no clue that they might be a carrier. And so that's another example of something that I brought up earlier, known as heterozygote protection. That allele is not acted upon, cannot be acted upon by natural selection. It can't be selected against and removed from the human gene pool eventually because it's hidden. It's being protected in the heterozygotes in the human population. So that's color blindness, and I kind of like this Punnett square because it uses the same symbols that we see in pedigrees to represent males and females and affected individuals versus non-affected individuals. And that's what we're going to be looking at on the next slide for hemophilia, a big old pedigree of the royal family. Rather extensive pedigree. And there are some things about this figure that I don't know exactly why they did it the way they did it. For example, you'll see instances like this where there's looks like this is Louis II is a father here, but there's no mother, but it's showing offspring from Louis II. So I don't know exactly how they put this thing together. But for the most part, you can see who's related to who and who is the offspring of who. And the first thing that you might realize and should realize is that the only ones that actually have full-blown hemophilia are males. And that's your clue that you're dealing with a sex-linked trait. Females can only be carriers, or are only carriers in this family tree here, in this pedigree. So this trait is inherited as sex-linked because only males are affected. That's your clue anyway. That doesn't mean females, it's impossible, but it's not likely. So just like color blindness is caused by any one of 3 genes, hemophilia can be caused by any one of 2 genes that code for blood clotting factors. So if these blood clotting factors are not functioning or not present in the blood, then that means the blood will not clot, and therefore you can bleed to death just from a small cut. These people also bleed or bruise easily, and they can also experience internal bleeding that can cause them to bleed to death internally. not a nice disease. And usually those males with full-blown hemophilia are not going to live long and probably will not reproduce. But there is an exception to that in this pedigree. So if you analyze this pedigree for how hemophilia got into the royal family and how this all started, as you trace back and trace back and trace back, you can see that it started right here with Queen Victoria. But notice that her parents didn't have it. Her father didn't have it, and her mother's not a carrier, and her grandparents, same thing. So where the heck did it come from? Well, she's a mutant. For some reason, the gene mutated in her, and therefore it started with her, and it got passed on through the royal family, and it originated with her. She's a mutant, like an X-Men. Unfortunately, that didn't impart on her any kind of superpower. It's negative. The other thing that can be observed here is something that you may have heard happens or happened in the royal family, and that is inbreeding. But the technical term for that, and that's the reason I'm bringing it up, is consanguinal mating. In other words, Queen Victoria and Prince Albert here were cousins, and that's known as consanguinal mating. We often refer to it as inbreeding. And really the only problem with consanguinal mating is an increase in the incidence of homozygous recessive individuals in offspring. It's more likely that two related individuals are both going to be carriers of recessive alleles, and therefore their offspring are more likely to be homozygous recessive and show those recessive traits, which are often, but not always, undesirable. So we'll leave it at that. So as we see in these sex-linked disorders, because the females have no idea they're carriers, and this is back in time when they had no idea how to do genetic testing or didn't really worry about or understand how traits like this were passed through the population or passed through families, the female carriers perpetuated the disease in the Russian, Prussian, and Spanish royal houses. So it spread throughout royalty in different countries. And then the last thing to note here, just interesting, is that Leopold was the only hemophiliac to have offspring. Usually a male that has hemophilia is not going to live long enough to reproduce, but Leopold here lived long enough to reproduce somehow. I guess he was real careful and didn't bleed to death before he could reproduce, but so he definitely passed it on to his daughter, Princess Alice here. And Princess Alice could have had hemophilia if her mother was a carrier. So if there was some inguinal mating here in the royal family, then that would have, and that's kind of an example of what I was trying to explain earlier, that it increases the incidence of recessive, homozygous recessive individuals in the population. And I think I just mistakenly said inguinal. I meant consanguinal. Inguinal's something different. That's like your armpit. That's an inguinal area on your body. So I'm sorry about that. Consanguinal. So all of these disorders are being caused by mutations in genes and therefore defective proteins. And this is another one, Duchenne muscular dystrophy. So dystrophin is the protein that is missing in those people or malfunctioning in those people that have Duchenne muscular dystrophy. So it's part of a protein complex that links the cytoskeleton to the surrounding basal lamina surrounding the muscle cells in your body. So it connects the cytoskeleton to the basal lamina. The basal lamina is extracellular matrix of collagen fibers and other proteins. And you'll notice that there's a link here between the two, the cytoskeleton being made up of microfilaments. Those are the ones that would be closest to the plasma membrane. That's where the microfilaments of the cytoskeleton hang out. If you'll recall, the other part of the cytoskeleton are microtubules, but these are microfilaments in this diagram. So there's a connection between the cytoskeleton microfilaments and the basal lamina outside the cell, and so that anchors the cell. And it's a complex of proteins. Dystrophin's the one that we're only going to really worry about, because that's the one where the mutation causes it to either malfunction or not be present. So that's what connects them. It's a long protein with numerous redundant coils that acts like a shock absorber. It's almost like a spring. So it acts like a shock absorber during muscle contraction. And if it's not there, without the support and protection of dystrophin, muscle cells are subject to damage and atrophy. And that's what causes the disease. Again, the disease is known as muscular dystrophy. So here's a normal boy next to a boy that has muscular dystrophy, and you can see that he hasn't developed as well, and his muscles are shrunken and weak. And that's what's known as atrophy. Atrophy is when muscles become weak. So Duchenne muscular dystrophy results from mutations in the DMD, Duchenne muscular dystrophy gene, that encodes the protein dystrophin. There are many mutations that cause the disease, and each one results in a frame shift. If you recall, that's a kind of mutation that shifts the reading frames of codons in the messenger RNA, and in this case, it produces a stop codon. And if you recall, when that happens, when there's a frame shift that results in a stop codon, or any mutation that results in a stop codon, we call that a nonsense mutation, because the production of the protein is going to abruptly stop before the protein is complete, or the polypeptide is complete. And so that's a nonsense mutation. That protein is nonsense, completely non-functional. So the DMD mutation, they're particularly common in hotspots, as they're called, in exons 44 through 55 and 2 through 10. Now notice we're looking at exons here. We're not looking at base pairs, because this gene is huge. So this is the normal messenger RNA, and 1 through 79 here represents the exons. And if you recall, exons are regions of the RNA that are kept, whereas introns are regions of the RNA that are cut out. So if you'll recall, during transcription of RNA from DNA, the RNA that is originally transcribed from DNA is pre-mRNA. That pre-mRNA then is processed, and through processing, the introns are cut out and the exons are spliced together. And so that's what we're looking at here. We're looking at a finished messenger RNA. It's gone through processing, and all the exons have been spliced together. And there are 79 exons that make up this protein. And it's a very long protein. It's actually the largest known gene, is this gene that codes for this protein. 2.4 million base pairs. 2.4 million base pairs long. That's a long gene. It being so long might even be one explanation for why it mutates so often. So here we're looking at a mutated messenger RNA, and as you can see, we hit a stop codon here in exon 44, and that causes that exon to not be finished, and all the exons following to not even be partially made. So this protein is completely incomplete. So it's not even going to, for example, you can just picture it being cut off right here, and therefore it's not even making a connection to the rest of the complex that connects it to the extracellular matrix. So it's completely non-functional. So loss of dystrophin function results in decreased muscle development and weakness and atrophy, as I already pointed out. And that's what it looks like. And eventually total loss of muscular function. So it's not a good disease. Just like hemophilia is not a good disease or disorder. I guess no disease is a good disease, huh? So the last thing we'll look at in this series is what's known as X-Inactivation. And this is something that probably you girls are going to find most interesting. So just to remind you, females have 2 X chromosomes and males only have 1, right? That's a huge difference between the genetics of male cells and female cells. So the problem here is that if both X chromosome genes were expressed in females, that would make twice as much of the protein as males. In other words, the more genes that are being expressed, the more proteins being made. And since there are 2 X chromosomes in female cells, if all the genes on both chromosomes were being expressed, that means that there would be twice as much protein. And that's not necessarily a good thing. Actually, it would be a bad thing. And those individuals would probably never even be born, because it's such a bad thing to have too much of any particular protein. The amount of protein in cells is very tightly regulated through that whole idea of transcription factors and so forth to turn genes on and turn genes off. So that's a problem. And the way that nature or evolution has overcome that problem is to turn off one of the X chromosomes in every cell in every female. And that's known as X-Inactivation. It's the random deactivation of one X chromosome in each female cell. And it was first discovered in 1961, just 2 years before I was born. And I know that seems like a long time ago to you, but not to me, by a British geneticist named Mary Lyon. So it's a relatively recent discovery. We had no idea before 1961 this was going on. And it definitely does not occur in males because if you think about it, males can't survive without their X chromosome. They only have one X chromosome, and if you were to turn off that one X chromosome in any cell, it would result in the death of that cell, and if it happened in a lot of cells, it would result in the death of the entire individual. That individual would never be born, basically, because the genes on the X chromosome are vital. Vital meaning you can't live without them. So the visible result of X inactivation are these things that we call bar bodies. Bar bodies are the inactivated X chromosome in females that condenses and attaches itself to the inside of the nuclear envelope. And that's what we're looking at in these images. So that's a male, that's a female. These are cells from a male, cells from a female. And the spheres that we're looking at here, the circles we're looking at, are actually the nuclei of those cells. So you can faintly see the outline of a cell membrane here. So what's most visible here are the nuclei of those cells. So those are nuclei, and you can notice also that inside the nuclei there are nucleoli, sometimes more than one nucleolus. And the bar bodies are being pointed to by these black arrows. So right here, that dot, that black spot there is a bar body. So it's a condensed, inactivated X chromosome attached to the inside of the nuclear envelope. Same here, same here. So you can look under the microscope at cells and determine whether they're cells from a male or a female by identifying whether or not there are bar bodies. And that could be useful, like at a crime scene. You could quickly determine the gender of a perpetrator or the victim from blood evidence under the microscope. You could just take some blood evidence, put it on a slide, look at it under the microscope immediately, and know whether or not you're looking at blood from a male or a female. Okay, by now you might be wondering why we're looking at a picture of a cat. Well, that's a calico cat. And calico cats, you may have heard even that calico cats, true calico cats, are only female. Only females can be true calico cats. Because in female cats, some of the cells have the X chromosome with the orange allele, and other cells have the X chromosome with the black allele. So only the orange allele is being expressed in those cells that have that X chromosome with the orange allele, and then the black is coming from cells that only have the X chromosome active that has the black allele. And so you can actually visibly see the result of X inactivation in calico cats. Now the thing that's really more interesting is that it's the same in human females. It's the same in all animals that show X inactivation. So basically, even human females can be considered maternal-paternal chimeras, because remember, you get one X chromosome from your mother, and you get one X chromosome from your father. And in some of your cells, your mother's X chromosome is being expressed, and in other cells, your father's X chromosome is being expressed. So that makes you a mix of your mother and father's, the expression of those genes on the X chromosomes. So that's what we call a chimera, a mixture. And just like in calico cats, this can be actually visible in some human females. This can cause different colorations in skin, for example, and other things. So that's a look at our current understanding of the X and Y chromosomes and the strange things that go on in heredity concerning the X and Y chromosomes.